Hello, everyone, and welcome to today's webinar, Gaining Multidimensional Insights into Cancer Heterogeneity, Co-Analysis of CTCs and ctDNA derived from a single liquid biopsy sample. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Bio-Rad Laboratories. To learn more, visit bio-rad.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. With that, I'd like to now welcome our speakers, Dr. Yoon-Tae Kang, Staff Scientist, Marketing Applications, Digital Biology Group at Bio-Rad Laboratories. Also joining us today is Abiodun Bodunrin, PhD, Application Scientist, Marketing Applications, Digital Biology Group at Bio-Rad Laboratories. For a complete biography on our speakers, please click on the Presenter tab. Dr. Kang, Dr. Bodunrin, you may now begin your presentation.
Thank you for a great introduction, Kaylee, and welcome to today's webinar. Again, I'm Yoon-Tae, and with my colleague, Abi, we are going to talk about these topics. So at first, general trend of cancer diagnosis and prognosis using circulating marker in terms of a liquid biopsy, and use of the more than 2 circulating marker for cancer heterogeneity studies. To do this, we are going to cover some of the research and technological innovations to make liquid biopsy possible, and in a later chapter, we will share our new protocol and demonstration of a workflow, mainly about how the recent technological breakthrough in this field can be applied to the circulating marker-based cancer studies. As you may know from lots of journal paper and news article in the cancer field, liquid biopsy is a new trend of disease diagnosis and monitoring. Unlike the conventional tissue biopsy that involves invasive removal of tissue sample to diagnose cancer and disease, liquid biopsy uses a much less liquid sample, such as blood, spinal fluid, and saliva. It is minimally invasive and low cost, and enabling repeated tests so that doctor can monitor their cancer individual's disease progression and tumor evolution from time to time. Also, liquid sample provides spot information at the time of sampling, so it is considered better for capturing cancer heterogeneity over conventional tissue biopsy. Let's talk about the circulating marker you can get from the blood sample. Blood is a great source of circulating marker, and actually it can contain many different biomarkers in the plasma and cellular fractions. So, for example, plasma contains extracellular vesicle called EV and protein, and circulating tumor DNA, RNA, and tumor-educated platelet called TEP. While the cellular fraction contains circulating tumor cells called CTCs, cluster CTCs, and immune cells, etc. So among those markers, circulating tumor cell or CTCs are living cells that are released from the primary tumor and considered the seed of metastasis. With the scientific advances in this field, we are getting a better understanding of the metastatic process, such as the EMT, which is endothelial mesenchymal transition. And this transition where the tumor cell loses its ability to be adherent and dissociate from the primary tumor and enter circulation, and eventually metastasize to the different organs such as bone, lung, and liver. The CTC can be successfully captured from the blood, and thus collection of liquid biopsy sample from cancer individual can serve as a good source of cancer disease and monitoring. These advances may be both logistical, because such as ease of the collection and serial testing, and biological, as these CTC may better reflect the whole body burden of the cancer, provide a comprehensive snapshot than the conventional tissue biopsy. And it is equally important that CTCs are ideal marker for both cellular and molecular studies, as well as actual functional studies to study cancer progression and metastasis. Another promising liquid biopsy marker that can give us information about the tumor is actually circulating tumor DNA, called ctDNA. ctDNA can be detected in plasma and other body fluids, and those are mainly derived from the dead or damaged cancer cells. This is a type of cell-free DNA, which is bigger category of those genomic DNA out of cells. Basically, using blood sample, the ctDNA can be isolated in the plasma fraction after a relatively simple centrifugation step, and this extracted ctDNA can be studied in terms of total ctDNA concentration and specific mutation analysis and methylation pattern analysis. Due to this easy access to the sample, ctDNA has been used in many minimal residual disease studies called MRD studies in either research level and actual clinical setup. To make liquid biopsy possible, we should be detecting scalable level of certain tumor marker for actual cancer studies. In a recent paper, researchers obtained a peripheral blood sample from over 2,000 patients with different cancers, and the first thing they did was to get rid of other blood cells, such as RBC and WBC, via centrifugation, and they also use a magnetic bead that can bind to the oral leukocyte, and finally, all the remaining cells were stained manually to see individual rare cell population. In this study, they specifically used two different terms, CTCs and CTECs, which is certain tumor-derived endothelial cell, specifically expressing endothelial marker CD31. And as you can see here, different cancer types have a different range of certain cancer cell numbers. And thankfully, this showed that 91% of cancer patients has at least one CTCs with wide range of numbers, supporting the feasibility of the recent trend in CTC-based liquid biopsy studies. For the ctDNA prevalence in liquid sample, I introduce my colleague Abi again, who is also an application scientist in Bio-Rad.
Thank you so much, Yoon-Tae. So previous studies have shown through the research the detection sensitivity of ctDNA in multiple cancer types, and also the mutational landscape reviewed by ctDNA profiling. The figure shown above shows that allele frequency of ctDNA mutations varied across different cancer types. For instance, the small cell lung cancer, SCLC, exhibited a high allele frequency compared with other types, whereas the lowest allele frequency was observed for cerebral glioma. This is possibly due to the blood-brain barrier. CTC and ctDNA have immense clinical applications. Analyzing CTC and ctDNA has opened a new vista in diagnostics and are arguably the cornerstone of non-invasive diagnosis. Blood samples can be drawn repeatedly to predict relapse or metastatic progression in patients. They can also be used to monitor the efficacy of therapies and understand potential resistance mechanisms. Before therapy, patients can be stratified to the most effective drugs, whereas after initiation of treatment, persistence in increases of CTCs or ctDNA indicates resistance to therapy, and this information may allow an early switch to a more effective regimen before the tumor burden is excessive and incurable. But can they completely replace tumor burdens in the near future? Speculatively for now, but over time will tell. So here we have a comparison of CTCs and ctDNA. The advantage to analyzing ctDNA has shown that it is relatively easy to retrieve from blood samples. However, the disadvantages to studying ctDNA include ctDNAs are short pieces of DNA, roughly an average of 150 kilobases. Each molecule will contain limited genetic information. Another disadvantage is that because they are no longer associated with a living cell, these molecules cannot provide information with regard to gene regulation of the original cancer cell, nor can information about metabolic status, processes of the cancer cell be discerned. Thus, a synergistic approach to studying both CTCs and ctDNA can provide greater insights into cancer genomics, status, progression, and monitoring. And that is what this webinar is all about. So what I'd like to present to you today is a new workflow for liquid biopsy processing that allows capture of both CTCs and ctDNA for examination of cancer heterogeneity. Tumors contain a heterogeneous mix of cancer cells. Tumor heterogeneity can be distinguished into spatial and temporal heterogeneity. Spatial heterogeneity is the uneven distribution of cancer subclones across different regions of the primary tumor. And temporal heterogeneity is the variations in the molecular makeup of a single tumor lesion over time. Clonal heterogeneity emerges when different sets of mutations and deletions drive different cancer cell clones, generating zones within the tumors that contain unique sets of cancer cells with defined genetics. Therefore, tumor biopsies or portions cannot provide a view of the entire landscape of cancer-associated genetic defects. Analysis of CTCs and ctDNAs from the patient's blood can potentially overcome this limitation and shortcomings and offer precise genetic information reflecting the status of all the cancer cells in the tumor in order to account for tumor heterogeneity. So looking at the figure before us, a zoomed-in view shows cells of different colors within a heterogeneous tumor. 3 biopsies are taken from different areas within the same tumor, and each biopsy reveals a distinct cancer genetic profile. Now, when we draw blood, on the other hand, the blood sample contains circulating tumor markers, CTCs, ctDNA, and extracellular vesicles. Whole genome or RNA sequencing can be performed on these markers, and the result is a comprehensive analysis of all cancer genetic profiles. The image emphasizes the importance of understanding cancer at a molecular level by examining both tumor tissue and blood sample. So why is identifying specific mutations in cancer important? The figure above shows the biological logic of new biomarker-guided clinical trial design in precision medicine. This was made possible by understanding the specific mutation underlying different cancer types. The essence of precision medicine is to explore the unknown relationship between drugs, targets, and diseases in the human body. The figure on the right shows the mutational landscape occasioned by ctDNA profiling. We have a heat map illustrating the top 20 most common mutant genes with color gradation that represents the mutational prevalence of each gene in different cancer types. The right bars indicate the distribution of different mutational types for each displayed gene. For example, we see the missense mutation as a large driver for a series of genes implicated in different cancer types. So here we have a slide showing an innovative approach that allows simultaneous evaluation of numerous markers and methods within a single blood sample. This is an exciting field in cancer research, as discussed earlier. On the left, we have an analysis that is consistent with CTCs, and on the right, we have a commensurate analysis using the ctDNAs. Combining CTCs and ctDNA in a single blood sample provides a comprehensive view of tumor dynamics. Researchers can boldly simultaneously access genetic alterations and functional properties. This multimodal approach enables early detection, it enables treatment monitoring, enables prognosis for predicting patient outcomes, and functional insights. I would like to pass it on now to Yoon-Tae.
Isolating multiple markers in one sample sounds straightforward and simple. However, there are not many commercially available and reliable systems to make it possible. So with optimization and novel protocol, you could finally do more comprehensive study using more than one circulating marker for cancer studies. And at the same time, you should also keep in mind that we need to isolate those circulating markers with non-biased ways and extreme sensitivity so that we can gather as much information as possible from the clinical sample you have. So I'm going to walk you through several recent technological breakthroughs regarding circulating marker isolation and high sensitivity analysis. So first of all, to isolate circulating tumor cell, there are two main approaches for the last 2 decades, which are label-dependent isolation and physical isolation. So label-dependent isolation uses antibody or specific biochemical molecule to target surface protein expressed on the CTCs. This was widely used previously due to its high sensitivity and high purity capture. But this method has a low throughput and can cause biased selection of CTCs out of all kinds of CTCs from the cancer individuals. Physical isolation actually make use of difference in physical properties of the cell in the blood, such as size and deformability and density. These methods include filtration and fiber-based gradient centrifugation, electrical field separation, and some of these combinations. These methods are usually high throughput and has an advantage of capturing heterogeneous circulating tumor cells, even though there is possibility in losing smaller CTCs. But more importantly, the first FDA-cleared CTC technology that uses anti-epithelial cancer adhesion molecule called EpCAM has a major drawback in wider use. The EpCAM expression on CTCs are very different depending on the source of CTCs from cancer individual. For example, if your CTCs are coming from myeloid or melanoma patients, there is high chance you losing those CTCs due to their low or zero expression of EpCAM. So in summary, many of previous CTC systems has lack of sensitivity and long processing time, low capture efficiency, and low throughput. Here, in order to gain more insights into cancer heterogeneity, specifically in CTCs, we use this label-free CTC isolation system called Genesis with cell select slide technology. This technology resolved many of previous challenges in CTC isolation by providing a fully automated isolation and enumeration of CTC from blood. This instrument uses cell select slide technology that can isolate CTCs without any pre-processing and labeling procedure that can cause potential cell loss and longer processing time. The key technology cell select slide is designed with 56,000 microchamber that trap CTCs, while 99.9% of blood cells, such as RBC and WBCs, are washed away. As the system is label-free, it can isolate EpCAM negative cell and mesenchymal CTCs, such as CTC from melanoma patients. It has a higher capture efficiency at many different cancer types over previous label-based methods, and the system automation allows users to reduce hands-on time, thus increasing overall sample throughput. And it also allows to create their own protocol and their own antibody staining panel. So it is very customizable and flexible, especially if you want to explore more about your target sample. The system supports 2 validated workflows for users, enrichment and enumeration. The enrichment protocol is basically to isolate and release the isolate cell for downstream analysis so that you can use these rare cell population for several molecular studies and functional studies, such as sequencing, qPCR, ddPCR, and cell culture. And the enumeration is automated immunostaining protocol after target cell isolation on slide, including cell fixation and permeabilization and staining. It is very powerful given that people in the CTC field spend significant time to stain and visualize captured cell to figure out what those cells really are. So we are going to cover these 2 system protocol options for 2 workflows, such as EGFR mutation detection in CTCs and dual CTC ctDNA isolation workflow we are going to talk about later chapter. And back to Abi.
All right. Thank you, Yoon-Tae. An instrument that plays a pivotal role in analyzing ctDNA in liquid biopsy is the ddPCR, which stands for Digital Droplet PCR. ddPCR provides absolute quantification of nucleic acids with high sensitivity and precision. It excels in detecting low abundance targets, even in complex backgrounds and in the midst of PCR inhibitors. In blood, where cell-free DNA, ctDNA, and CTCs can exist at low levels, ddPCR sensitivity is extremely crucial. As mentioned in the previous slide, the key technical advantages of ddPCR include absolute quantification in copies per microliter. We do not have a standard curve, and because we read at endpoint, there is less worry about PCR efficiency and increased resistance to inhibitors. This is especially important on degraded clinical samples. ddPCR has high precision and reproducibility, allowing ability to discriminate small differences in gene content. Its high sensitivity enables detection of rare events like cancer mutations and viruses. This is fundamentally due to titrating out high copy background, effectively increasing the signal to noise ratio. In the figure above, samples are loaded on wells in the cartridge. The droplet generator oral is added into a different compartment and placed in the droplet generator, which uses specially developed reagents and microfluidics to partition its sample into 20,000 nanoliter sized droplets. As shown, target and background DNA are distributed randomly into the droplets during the partitioning process. ddPCR also has an advantage in detecting rare mutations amidst normal DNA. In the figure above, we see ddPCR being used in targeted mutational screening, minimal residual disease and monitoring, identifying cancer subtypes, optimizing drug treatment plans, monitoring residual diseases, studying tumor evolution, and detecting rare tumorigenic mutations, often down to 0.01% or even lower. ddPCR enhances our understanding of cancer genetics. In the figure shown, we see the partitioning effect, which is a hallmark of ddPCR technology and has an important impact on the sensitivity and specificity of a PCR reaction. For applications that are reliant on measuring a low abundance rare mutant allele in a large excess of wild-type DNA, partitioning the sample into droplets increases the sensitivity by orders of magnitude by effectively diluting away the background. So here, we show some advantages that ddPCR has over NGS technology. Ultrasensitivity, which is ddPCR excels in detecting low abundance ctDNA levels, as can be seen in the chart above. We also have absolute quantification, which provides precise allele quantification. And it's rapid and economical. It offers a cost-effective solution for ctDNA analysis. So now we go on to talk about a model sample study that we carried out and the workflow that was used. So in the workflow being introduced subsequently, we demonstrate a protocol that shows CTC enrichment and EGFR mutation detection, and also the dual isolation of ctDNA and CTCs from the same sample. In workflow 1, we show CTC enumeration and EGFR mutation detection encompassing CTC isolation, release, and CTC genomic DNA extraction and mutation analysis. In the second workflow, we show co-isolation of CTC and ctDNA from the same sample for comprehensive liquid biopsy. Please note that if workflow 2 is intended, it must be preceded by the CTC enumeration and enrichment steps. The two workflows was facilitated with the Genesis cell isolation system and droplet digital PCR, along with optimized blood component separation and cell-free DNA enrichment steps. After sample preparation, the Genesis system with cell select slides enriches CTCs based on size and deformability, and the enriched cells can be recovered for subsequent downstream analysis. Concurrently, ddPCR can be utilized to interrogate specific loci within cell-free DNA to determine the presence of a range of DNA aberrations present in ctDNA, including single nucleotide variants and insertions and deletions. I'd like to pass it on now to Yoon-Tae.
Thank you, Abby. So, in this first workflow, we wanted to detect different types of EGFR mutations in CTCs. So as a model CTCs, we prepare 3 different lung cancer cells, A549, H1650, and H1975 that has a EGFR wild type, EGFR exon 19 deletion mutation, and EGFR point mutation such as A58R and T790M, respectively. So we first spike the low number to high number of cancer cell into healthy donors' blood to mimic lung cancer individuals with different cancer stages. And then we follow these 2 sequential workflow, the part 1 rare cell enrichment and part 2 EGFR mutation detection. So please note that if you have a clinical sample from cancer individual, you could use that sample without spiking any cancer cell into blood. In this workflow, we use a model sample with spike cancer cell, and we isolate cancer cell following the Genesis instrument enrichment protocol. And later, these enriched cells were subsequently lyzed, and then genomic DNA from enriched cancer cell undergone ddPCR-based EGFR mutation detection. Detail. Again, blood sample, either from clinic or model sample you prepare, can follow this second step, rare cell isolation. Typically take less than 1.5 hours. And Genesis system automatically release the capture cell, and this cancer cell containing sample is further used for the total gDNA extraction. We recommend user choose DNA extraction reagent for whole blood among several options out there, because that can give you a higher total gDNA extraction and eventually better result in ddPCR. In this study, we used DNA Joule BD reagent. And finally, this sample from step 4 is compatible with conventional ddPCR workflow with your choice of mutation detection assay. To evaluate the capturing performance of the system, we count the capture cell number in the system before the enrichment. And we show that 78 to 90% of the initial cells we spiked into blood were captured using Genesis cell select system. As a cancer individual with different cancer stage has a different range of CTCs in the blood, we also evaluate the sensitivity of the system with low to high cell number and showing a great linearity and sensitivity, as you can see on the right graph. So this system isolation technology is suitable for a wide range of cancer cell types and CTC number in different cancer stages. For ddPCR, you use the extracted DNA sample from the post-CTC isolation sample using DNA extraction reagent. Here, we specifically aim to target certain EGFR mutations in the CTCs. Bio-Rad already offers a wide variety of mutation screening kits and detection assays for cancer markers you might be interested in for ddPCR. So here we specifically use exon 19 deletion screening kits and 2-point mutation assays, followed by general protocol of ddPCR and assays. And making ddPCR reaction mix and generate droplet, perform PCR, and read and analysis. And back to Abi.
Thank you, Yoon-Tae. So now I'm going to walk us through results that we got for enriched cancer cell and ddPCR mutation assays. In the figure above, we show results of profiling EGFR DNA mutation analysis in circulating rare cells using the Genesis cell isolation system with cell select slides and droplet digital PCR. As mentioned previously, EGFR mutation is critical for determining the most effective treatment for patients with lung cancer. We show a 2D plot of lung cancer cells spiked in blood followed by DNA extraction, which shows the H1975, which has the mutation EGFR T790M and L858R, We also showed cells with H1650, which shows the exon 19 deletion, and the A549 cells, which are the wild type, with different concentration of cells collected from the rare cell isolation steps using the Genesis system with cell select slides. The cells, as we can see, are numbered from 100 cells, 400 cells, and 1000 cells, and these were recovered and we ran the mutation detection assay on them. High sensitivity of the ddPCR mutation detection assay provides precise quantification of T790M, L858R, and the exon 19 deletion mutation in the presence of wild type and mutant DNA. The black cluster on the plots represent the negative droplets. The green cluster represents the droplets that are positive for wild type DNA only, in this case, the A549 cells. And the blue cluster represents the droplets that are positive for mutant DNA only. So this is the second workflow of the protocol that was introduced earlier, showing the co-isolation of CTC and ctDNA from the same sample for comprehensive liquid biopsy. We went through the following steps. Step 1, model sample preparation and plasma separation, in which the blood sample is optimized and pre-processed for the dual isolation. Step 2 is the CF cell-free DNA extraction and ctDNA mutation analysis with ddPCR and bespoke mutation detection assays from Bio-Rad. Step 3 is this rare cell enrichment and enumeration. So reiterating an overview of our workflow, lung cancer cell lines, the A549 and enzymatically fragmented cell-derived ctDNA from a company was spiked in blood to model CTC and ctDNA respectively. Plasma separation was tested using two common methods highlighted above. Cell-free DNA extraction was tested using two readily available cell-free DNA extraction kits, the magnetic bead-based kit and the silica affinity-based kit. So I'd like to pass details on to Yoon-Tae.
Again, using a blood sample containing more than one markers, here CTCs and ctDNA can be separated from cell fraction and plasma layer respectively. And plasma sample containing most of ctDNA and cfDNA can be followed by general cfDNA extraction kit protocol. And for ddPCR-based ctDNA mutation assay, we could follow general ddPCR workflow similar to EGFR mutation detection in CTCs we covered previously. And simultaneously, cell layer containing rare cell, such as CTCs, is undergone general rare cell isolation protocol using Genesis. These CTCs can further be stained using enumeration protocol, or if you also want to see EGFR mutation in the CTCs, you can simply follow the previous CTC mutation detection workflow we covered previously. And in order to evaluate how our system and workflow are compatible with different use cases, such as sample collection type, plasma extraction method, and cfDNA extraction kit choices, we first evaluate CTC capturing performance with 5 different sample collection tubes. So as you can see here, most of the blood collection tube on market are compatible with 2 workflow Genesis system offer, enrichment and enumeration, with the marginal difference one another. And then we also specifically evaluate a cfDNA recovery that is inside the range of 120 to 220 base pair in different scenarios. Using the model sample initially collected in the EDTA and STRAC tubes, we showed very similar results of total cfDNA extraction performance from magnetic beads-based cfDNA extraction kit and silica gel membrane-based cfDNA extraction kit. And also, we evaluate any cfDNA recovery difference between 2 plasma extraction methods we explored here, which is Vicor-based and 2-step centrifugation. And our data show similar cfDNA recovery that is over 70%. And this time, we demonstrate an enumeration protocol using this sample containing CTCs and ctDNA. As you can see, if you follow our optimized protocol for cell layer separation and Genesis-based CTC isolation, potential loss of cell you have might be less than 10%. Even after plasma layer separation using vesicle or general two-step centrifugation, more than 70% of cancer cells you spike are actually captured. Using a standard CTC staining panel, including cytokeratin and DAPI, as well as negative marker, such as CD45 that is expressing on the white blood cell, we confirm and identify CTCs after automation of the CTC enumeration protocol using our Genesis system. And finally, for ctDNA, using ddPCR platform and mutation detection assay specific to T790M point mutation, we demonstrate an exclusive positive droplet detection only from plasma sample with ctDNA we spiked. As you can see in the first two graphs on the left with the cfDNA extracted using magnetic beads-based kit, healthy donor plasma doesn't show any positive droplet. However, the sample with ctDNA spiked showed EGFR T790M positive droplet. And we saw the similar trend in the another two graphs on the right side, using the sample with silica affinity-based kit. From this study, we found that these two market-available cfDNA extraction kits are compatible with general ctDNA detection with marginal difference using ddPCR. Again, this is a good demonstration that high sensitivity of ddPCR mutation detection assays provide a precise quantification of EGFR T790M mutation in the ctDNA. And final summary is back to Abi.
Thank you very much, Yoon-Tae. So in summary, we have demonstrated a label-free viable CTC isolation protocol. We've shown sensitive detection of cancer mutations and circulating markers, the CTCs and the ctDNA. And we've also showed a streamlined workflow for cancer discovery, facilitating in-depth study of diseases. So I would like to show here some of the products that we used. We have the Genesis system with cell select slides, as we mentioned previously, which automates the capture of rare cells for recovery or on-slide immunostaining. We have the ddPCR technology, which is Bio-Rad's unique digital PCR technology. We have the ddPCR QX200 and the QX600. We have the cancer mutation assay kits, which is what we used for this study. Next slide. So find our recent protocols and app notes on profiling EGFR DNA mutation analysis in circulating rare cells using the Genesis system and ddPCR. We also showed the dual isolation and profiling of circulating tumor cells and circulating tumor DNA. And thirdly, we have a multiplexing mutation detection using our newly launched 6-color ddPCR instrument, where we looked at detecting KRAS, BRAF, and EGFR mutations. You can go to Bio-Rad.com and simply type these bulletin ID numbers at the bottom of the screen, and you can get these white papers freely. Everything that we've done in this protocol that we've shown was also used, can be found in any of these bulletins. You can find out more information on the Genesis system and ddPCR by following the link shown above. This concludes our presentation, and we are happy to take any questions that you might have for us. Thank you.
Great. Well, thank you so much, Dr. Kang and Dr. Bodunrin, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. Our first question here is for Dr. Bodunrin. This question asks, you showed how to detect EGFR mutations in your presentation. However, our lab is working on colorectal cancers. Would the workflow be different for colorectal or other cancer types? And what things would I have to consider?
Okay, thank you so much for that question. The protocol that we've shown, the workflow that we've shown for CTCs and ctDNA, they are very similar as long as CTCs in your cancer types are in the range that our system supports. We have many other mutation assays and kits, such as the KRAF G12C. We have the Bio-Rad V600E, and so many other ones like that, that encompasses other cancer types with different mutational landscapes. So there really should not be a problem with testing out colorectal cancer, pancreatic cancer, breast cancer, as long as those mutations, you can actually get the assays for them.
Great, thank you. All right, Dr. Kang, coming back to you for another question here. Because ctDNA is derived primarily from dead cells and CTCs are alternatively derived from viable primary cancer sites, are there any differences observed between the data derived from dead versus living cells?
Yeah, okay, that is a very good question, and clinicians and scientists like us are still investigating it. So for clarification, source of ctDNA will be various, including primary cancer and dead cell and dead circulating tumor cell as well, undergone apoptosis and necrosis. While CTCs are mainly coming from the primary tumor site. So for example, if any anti-cancer treatment is working successfully, but a patient's cancer is mainly resistant to that treatment, this therapy resistant tumor cell don't undergo apoptosis, and there is not much ctDNA you can use for analysis. So in this case, CTC might be a good source to analyze any genomic change during this treatment or something. So again, it's still developing area. So hope it answered your question.
Thank you. Now, Dr. Bodunrin, can you comment on why ddPCR was used for this workflow application versus qPCR?
Thank you so much for that question. As we've said in the previous slides, that ddPCR has obvious key technical advantages, one of which is absolute quantification in copies per microliter. Another advantage that it has over qPCR is that we do not use a standard curve for ddPCR analysis. And because we read at endpoints, there is less worry about PCR efficiency. And, ddPCR can be more tolerable to PCR inhibitors, which is not available for qPCR. Thank you.
Thank you. Great. All right, another question for Dr. Kang here. Since CTCs are very rare in circulation, how do the different single cell isolation methods compare with regard to capture efficiency? Also, how do the methods compare with regard to their gentleness of cells during the process? For example, are any methods better at preserving the native biology of the individual cells, which is essential for examining gene expression?
So again, there is mainly two different CTC isolation approaches out there, which is physical-based methods and label-based methods. And then physical-based methods are considered gentler, so that they can preserve the native biology of individual cell, actually. And another aspect we should think about here is heterogeneity of CTCs. So if your main goal is just gene expression analysis, the viability would be not super critical. However, physical-based method that is usually label-free can capture many potential CTCs, mesenchymal, and epithelial, etc. And that's going to give you more in-depth capture of cancer individual disease status.
Great, thank you. And I want to thank our audience for all these great questions. We do have a few more minutes here, so we'll continue with a few more questions. This next one is coming back to you, Dr. Bodunrin, and this one asks, can you discuss more about the mutation detection capabilities of the QX200 instrument and how it compares to the QX600?
Okay, thank you so much, Kayla, for that question. QX200 and QX600 are both of the droplet readers that we use in Bio-Rad, or that is available in the market. The QX200 has a two-color system, which is the FAM and the hex, while the QX600, which was just released about 2 years ago, I think, has a 6-color system. So it actually detects 6 targets. We have the FEM and the hex channel, we have the Sci5 and Sci5 channel, and we have the ROX and the ADO channels. One of the advantages that the QX600 has is that it has a faster turnaround time, and you can operate this at lower cost. And you also get to use less of your target compared to where you're going to be having a two-color system where you have to use more sample. Now, the major advantage that the QX600 has over QX200 in the bulletin that we talked about earlier, where we have the multi-mutation detection multiplexing capabilities, in that particular bulletin, we're able to, detect 6 targets as compared to 2 targets for the QX200. We're able to detect all in one sample the BRAF V600E, the KRAS G12C, EGFR T790M, EGFR L858R, and the deletion mutation. And we also included, as a last bit, the wild type detection of the EGFR. So 6 targets with just one sample in one well compared to, having to do this in 3 wells with three different samples for the QX200. I hope that answers the question.
Yes, thank you.
Sure.
All right. Now, Dr. Kang, do you observe any difference in workflows or results if whole blood versus blood minus plasma samples are used?
Yeah, actually, yes. And one of the slides I was presenting, showed that result actually. So PBMC separation, from the whole blood might cause some cell loss during pipetting and sample moving actually. Yeah, but thankfully using Genesis system and streamlined protocol we generate, we lost less than 10% of cancer cell we spiked for this evaluation. So I would recommend, practicing this pre-processing step if you haven't tried it before. So before you actually, deal with your clinical sample. So that's my observation.
Great, thank you. Now it looks like we have time for one more question here, so we'll wrap up after this one. And Dr. Bodunrin, this is coming to you. The data you presented showed that CTC's DNA had EGFR mutations, while ctDNA had DGFR mutations. Do these results hold any clinical significance?
Yes, thank you, Kayla, for that question, and whoever is asking that question in the audience. So it has serious clinical implications in the sense that EGFR mutations in ctDNA are a common biomarker in NSCLC diagnostics, and the presence or absence can shape the treatment of small cell lung cancers strategies and influence patient prognosis. Additionally, analyzing CTCs from the same sample can shed light on functional cancer studies and drug susceptibility passes. So that's almost like having a double whammy where you're analyzing 2 different things from the same sample. That's the import of the clinical significance of that. Thank you.
Great. Well, thank you. And I want to thank both of you, Dr. Kang and Dr. Bodunrin, for your time today and your important research. As this does conclude our session today, I'd also like to thank LabRoots and our sponsor, Bio-Rad Laboratories, for underwriting today's educational webcast. Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.